Hi all, I'm going to try something new today. I'm using a white paper background as requested. And I'm going to go over the Meselson Stahl experiments. These are the experiments that show that DNA replication is semi-conservative, meaning that you use one parental strand as a template to make a daughter strand so that each new molecule of DNA has one old strand and one new strand. So in the Meselson Stahl experiments, what they did is they labeled DNA with an isotope of nitrogen and look to see what happened over time. So if we just start out with 2 test tubes, we're going to grow our bacteria in here, and then we're going to see what happens. So here's our bacteria growing, and one of them we can label with an isotope of nitrogen that's heavy isotope, and one of them we can label with a light isotope of nitrogen. If we run these out on a density gradient, so we basically spin them in a cesium chloride gradient or whatever, what we see is that for the 15N we have a single heavy band, the 14N we have a single light band. So these are DNA bands. Okay, so this would be the light band, which represents 14N, and this would be the heavy band, which represents DNA that was made with heavy nitrogen. Okay, so this is what happens on the density gradient. Okay. So what Meselson and Stahl did is they actually grew up their bacteria in the heavy nitrogen, and then for one generation they moved them to the light nitrogen. Okay, and when they did this, they got a new result. So one generation, okay, and they were looking for some different possibilities. So possibility number 1, this is if we have what's called conservative replication. And in conservative replication, in this case, we would have a parental strand, which I'm going to draw in dark blue. Okay. A parental strand would then give rise to the same thing as itself, plus a brand new daughter strand. I'm going to draw the daughter strand in lighter blue. So this is conservative because we've conserved the original piece of DNA, this double helix of DNA, and then we've just made this whole new daughter strand. Now, if they saw this in their density gradient, what they would expect is that this molecule here would give us the heavy 15N band, and this molecule here that's hydrogen bound to itself would give us the light 14N band. Okay. The other possibility would be semi-conservative replication, and we all know that this is actually the way that it works. In semi-conservative replication, the parental strand of DNA splits in 2, and each of the 2 strands is going to serve as a template for daughter strand synthesis. Okay, so that after the first generation, each DNA molecule is made of one old strand and one new strand of DNA. So if we look at the density gradient for this, what we see is we see a mid-band. So this mid-band is right in between the 14N and 15N bands. Okay. Now they actually did consider one other possibility when they were thinking of this, which is the dispersive model. And in the dispersive model, oops, oh, that's the wrong color blue. In dispersive replication, the old strand would get used in bits and pieces. Okay. I don't know if I can actually draw this one, but I can give it a shot. Here we go, that's not so bad. Okay, so the old strand gets used in sort of bits and pieces to make the 2 new strands. I know exactly how I'm going to draw this. I'm going to draw it like this and then just draw the dark blue over it. There we go. Okay, so it gets used in bits and pieces, and so each strand of DNA is made up of old and new DNA. Now, if we look at the density gradient, though, this one is actually going to give us the same result as above because each strand is made of both old DNA and new DNA. So it gives us this middle band that has both the 15N and the 14N DNA in it. Okay. So what they did is they ran a second generation of this. So here's our first generation. And then if we look at our second generation of the density gradient, in the second generation under the conservative model, we would expect that we maintain this strand here. So this one gets maintained, it goes on to the next generation. And this one, we're going to just basically have two light versions of it since we're growing up a second generation in 14N. So we would end up with two of these and one and two of these. So we would have one light strand here, two light strands here, and one heavy strand here. So after the second generation, we would end up having the same 15N band and then a 14N band that had more DNA in it. Under semi-conservative replication, we would expect that each of these two strands would split into two so that we would keep one old piece for two of the daughters, and then this new piece here, this 14N, would be the template for the other daughter strand. So in this case, our second generation, I'm going to draw it a little bit over here just so I have room for it. Our second generation would still have this middle band here, the one that's made of 14N and 15N. And then it would also have these two light bands here. So we'd also have a light band. So we'd have 14N and then 14/15N. For dispersive replication, we would expect to see the exact same thing happening in the next generation. So each of these two mix and match strands would still be mix and match. And so in our second generation, we would still expect to see this sort of one band, only it would be even lighter than the first generation, since we grew a second generation in the light DNA. So this would still be, 15N, 14N hybrid for every strand, only now with more 14N than 15N. So what Meselson and Stahl actually saw, they actually saw this pattern here, where the first generation, they got this single band that was right in between the two original density gradient bands. And in their second generation, they got two distinct bands, one with this hybrid density and one with this lighter density. And so that confirmed that the semi-conservative replication model is the correct model for DNA replication.